Great. Good morning. My name is Joseph Mazar. I'm a research scientist here in Orlando with Nemours, obviously. I've been with Nemours now 7 years. I work under Tammy Westmoreland as the MD PhD PI for our lab here. We literally established it from scratch. When she arrived here, actually, I was working at the Sanford Burnham Research Institute, and so she wanted the collaboration to get her lab started. And I stepped in, and we hired some staff, got the lab running, and got our first paper published in 2 years. So we were very happy with that. And as a consequence, then she hired me to become full-time working here at Nemours. And we moved over to the Burnett School building in the UCF Medical School. And I guess the rest is history. So today, actually, I'm going to talk to you about one of our major projects, and this has actually been ongoing about 5 years or so. When we first started with the idea, it was more just sort of whim, just to sort of see what would happen, and it took off so beautifully that it became sort of a major panel of all of our research. So our background has always been on pediatric cancers, neuroblastoma, and actually a few others as well that we've looked into, but this has been really our focus. So today I'm going to talk to you about this project. Zika virus confers a dramatic survival advantage to mice with neuroblastoma. And we've gone through the entire progression of in vitro to in vivo, and now we're really sort of focusing on the preclinical trials for it. So I'll give you a background in all of the material over the years, sort of a brief amount of material. So a little background on neuroblastoma. It accounts for 8% of all malignancies in children. It's the most common solid extracranial malignancy in children. Most commonly within 40% present within the first year, usually within the first year to 3-4 years is when you see the majority. About 1 in 100,000 children under 15 acquire it, 1 in 7,000 births, 700 new cases a year. But really the most important statistic is this one. It accounts for 15% of cancer-related deaths. So despite only being 8% of malignancies, it's actually 50% of deaths, which really just sort of argues the urgency of this cancer. Now, amongst neuroblastomas, you have low risk and median risk. And intermediate risk. And these largely, so some of the low risk, they self-resolve. Actually, there's no therapy that's even needed. But between the two of them, about 90% plus survival rate. So this isn't really the concern. The concern is with high-risk neuroblastoma. And with these, the survival rate's only about 40 to 50%. So most of these children will pass. And that actually accounts for half of all cases. So high risk is actually slightly more than, it's about 52% of all cases are high risk. So this is where the real threat for this disease is. Okay, so when we look at the sympathetic nervous system, this is where neuroblastoma generally tracks. So it is a disease of neurons, neuroblasts, that do not properly develop into neurons. Okay, so it is not intracranial. It's actually tracks with the spinal column. The majority are generally the midsection, and ironically or strangely, about 40% track specifically to the adrenal glands above the kidneys. Okay. So our approach has involved, I would say, a bit out of the box. And it really just started around 2015 with the Zika outbreak. And there was a paper that came out in 2016 that indicated, strangely, that in these fetal cases of Zika, that the cells that were sensitive to the virus were these developing cells, but not the mature neurons. So for a fetus, the good majority of the brain tissue is in development process, and that's why they're so sensitive. So a little background on Zika virus. It's a member of the Flaviviridae virus family, so the same as dengue fever, yellow fever, Japanese encephalitis, that sort of thing. It's a single-stranded positive sense RNA, which means basically it acts like a messenger RNA. So it gets into the cell and it can translate almost immediately. So it's rather rapid. Most cases of Zika, though, are mild. So it's about 80% asymptomatic in humans, and it's probably much higher than that because many cases that are asymptomatic, you won't even go to a doctor to get diagnosed, so you never even detect it. So the remaining 20% cause a mild form of fever. It's really more like a cold, It's self-limiting, not chronic. Once you clear it, which is about 8 to 10 days, it's gone forever. And in fact, the irony is that most cases in human beings don't even elicit a significant antibody response. It's eliminated prior to antibody completion. Okay, so previous research has suggested that it could act as an oncolytic, and there's actually a number of groups that have done this in other cancers like glioblastoma, glioma, and so on, and other neural cancers. But we specifically looked at neuroblastoma. And our first paper actually, what we were able to discover was that a cell surface receptor, co-receptor called CD24, appeared to be essential for permissiveness of the virus. Okay, so some background on our first experiments. So we started this with the idea that if a developing neuron, a neuroblast, was sensitive to the virus, what would happen if we introduced the virus to a cancer that evolved from a developing neuron? In other words, a neuroblast that never developed into a neuron, it became a neuroblastoma, would it still be sensitive? And so we screened a couple different strains, and these are just two examples. So the first we have is IMR32. It's a MICAN amplified pre-treatment diagnostic neuroblastoma. So these are high risk. So MICAN is a critical biomarker for high risk in neuroblastoma. If you're MICAN amplified, it is automatically considered high risk. And what we discovered is when we introduced the virus, the IMR32s, which are the pretreatment, were hypersensitive and completely eliminated. But SKNAS, which is a post-treatment, it's actually a very late stage metastatic neuroblastoma. It originally had some sensitivity to the virus, but ultimately it recovered. Okay, so we got some killing, but then it recovered and the culture was intact. So I did some screening, next-gen sequencing, quite a bit of work, and that was mostly manual, not algorithm. And I stumbled upon a marker called CD24. And so when I screened CD24 expression, we found that SKNAS did not have appreciable expression of it. In fact, it was dramatically low. By qPCR, it's a CT of about 32, whereas the others are about a 22, 23. So it's almost 8-9-fold difference in CT. So I also did some fluorescent labeling after we added virus, and we could see here that with IMR32, we could easily detect envelope protein of the virus being produced in infected cells. But with the SKNAS, really just very little presence of virus. But when I reintroduced CD24, so I literally cloned CD24 from IMR32 and then exogenously expressed it in the SKNAS. And it's not really overexpressed because I never could get a CT as high as in the IMRs. It's more like a 26. So it's about maybe 12 to 15% of the expression in the sensitive strain. And lo and behold, that appeared to be more than enough. And now suddenly these cells were sensitive. You could detect virus permissivity, and the cells were killed in about 3 days, which is basically what we see with the sensitive cells. So I could make the cells sensitive simply by introducing a exogenous expression of this this gene. And here I'm just showing two different clones, variant 1 and variant 7. Okay, so we screened a whole bunch of these neuroblastomas, and every single one of them was sensitive except for this SKNAS. All of these, I should mention, are high-risk neuroblastomas. Some are pre-treatment diagnostic isolates, some are post-treatment recurrent isolates, and some are MICAN amplified, some are not. So we just tried to do an entire array of cell types. And basically, there was no factor that we could detect that made any difference, really, except for CD24 expression. So there you can see SKNAS as being really the only one that actually recovered. Okay, so the effect of CD24 on Zika infection. So again, went ahead and continued testing on CD24, and we did knockdowns. So this is just transient over 48 to 72 hours. Got about 70% knockdown, and then infected cells. And what we discovered was we really couldn't produce what's called NS1. So NS1 is non-structural 1. It is a Zika viral protein. It is not present in the virion. It's specifically and only present in permissive cells. So once the virus infects, this is the first structural gene that gets produced that is present inside a host cell, but never appears in a virion. So NS1 is a clear indicator that a cell is going to be permissive to viral infection, and ultimately it's lytic. It'll kill it. And so when we knocked down CD24, we could drastically decrease NS1 production, and it actually delayed viral killing from 3 days to about 5 to 7 days. And that was just with the single knockdown. So what we figure is that CD24 expression was recovering, and that was actually allowing for killing, but just delayed expression. So we did the opposite, which is, again, expressed it exogenously. So here you can see those variants, variant 1 and 7 again. And lo and behold, these are stables, by the way. When we did that, lo and behold, now we get NS1 expression within 24 to 48 hours. So very rapidly and consistent with what we've seen with sensitive cells, you introduce this gene, and now the cells are sensitive. They're expressing NS1. They're producing virus and virions and titer as a consequence, and the cells are getting eliminated in about 3 days. Okay, so this was some of the in vitro work that we did. And so now I'm going to turn to the in vivo work, and this is a little bit more recent. We've been doing this for, I think, about 3 years, all sorts of permutations, and I'll go through a few of them. So here we have our IMR32, that pretreatment MICN amplified, and below the SKNAS. So when we started this, we wanted to determine, could we determine an optimal dose of the virus to introduce that would have effectively an optimal effect on tumor elimination. And after a few tries, we stumbled upon a concentration of about 2 times 10 to the 6, so about 2 million virus, and we introduced this once. Mind you, I should point out all of our studies, the tumors, when they're introduced, we allow them to grow to be about 300 millimeters cubed. So that is the FDA standard for pretreatment trials for tumors. And so we wanted to maintain those standards. So at 300, we generally see that the cells have been grafted, and usually if you can establish that value, it will persist and will eliminate the mouse. So it's basically a guarantee that the mouse is going to die. And so at 300, we introduce a virus a single time, and then we wait and see what happens and measure. And so what we found with IMR32s is within 7 days, we could get total elimination of the tumors. We maintain these mice another 4 weeks, and we do not get recurrence. With SKNES, it was a bit surprising because the cells in vitro were rather resistant to viral killing, and in fact typically recovered. And so we sort of expected that might happen here, but it didn't. In fact, actually, we got elimination of tumor mass, 56% below start. But what was interesting is when we resected those tumors, there was no tumor mass left. It was just scar tissue. So the tumors themselves had been eliminated, and the region had just filled in with mouse interstitial cells. But what was interesting is when we screened for CD24 expression in the IMR32 tumors, we found there was a huge range of CD24, sometimes up, sometimes down. But generally speaking, statistically, it didn't really change much. With the SKNES, we were rather shocked to find expression had increased 30-fold on average. So that's not 30%, that's 3,000%. So this is about 5 to 6 CTs at least on average, sometimes higher. So expression went from about a 32 to about a 25, 26 CT by qPCR. And that boost appeared to make the cells suddenly sensitive, the tumors sensitive to viral killing. And so we see viral killing anytime we introduce the virus. I should point out, by the way, we introduce the virus by direct injection. So this is not by IP or by intravenous. We inject directly into the tumor and the bed around the tumor. So we continued with a time course study. So we wanted to have a little bit more detail. And what we found was that depending on the tumor, the first time point, which would be around day 2 to day 3, day 4, you usually saw suppression of tumor growth almost immediately. And then it diminished considerably after that. Again, that's after a single dose. What we also looked at was shedding. So that's very, very important from a clinical perspective because we need to know where the virus is going in the host. And what we found was with both IMR32 and SKNAS, the shedding of the virus, even though the tumor could produce 10 to the 6, 10 to the 7th virus, shedding was never more than about 10 to the 4th at the most, and it diminished with time. So usually by day 7, so and that's IMR, and day 10 for SK. So the reason those are important, that's the day at which basically loss of tumor mass was completed. So it completed its cycle, and the virus is often cleared at that point. And we can see here by shedding that we're at 10 to the 1, basically. At any other organ in the body. And that's important because virus is typically going to be found in the kidney or liver. With neuroblastoma, it often can transfer to the brain. Zika has been known, obviously, to travel to the brain. There's some evidence it can breach the blood-brain barrier, but as we can see here, that's not really happening. Now, one of the common responses we'll get is, well, this is a mouse model, so why would Zika do that in a mouse? Actually, it's been shown that mice are, in fact, a permissive host for Zika. It's actually been shown that you can get both mosquito to mouse and mouse to mosquito transmission. But what's interesting is that mice that retain type 1 interferon response eliminate the virus and are asymptomatic. We've even done this with NSG mice that lack T cells, B cells, natural killer cells, macrophage and dendritic activity, and Zika does absolutely nothing. But there was some studies that actually published in wild type mice where they knocked out type 1 interferon and the mice succumbed in 7 days. So the virus is entirely capable of infecting the mice, but their type 1 interferon response is eliminating it. And that's what we're seeing here. We believe that's probably the reason why in humans it's about 80% asymptomatic, is our type 1 interferon is eliminating it, and so ultimately you have no symptoms at all. Okay, so we also looked at susceptibility to Zika based upon CD24 expression. So this is a matching patient pair. So SMS CAN and SMS CAN-R. CAN is the pre-treatment diagnostic, CAN-R is the post-treatment recurrent. And so we wanted to know within the same patient sample, was there a difference that could be determined from pre-treatment to post-treatment? And generally speaking, not really. The tumors get eliminated, and generally speaking, any remaining mass is usually just scar tissue. And again, we retain these mice for 4 weeks following, and we don't get recurrence. And in these cases, their CD24 expression is rather high to begin with. So statistically, really no difference between in vitro and in vivo. It didn't really change, but they were sensitive either way. So that was our 4 different models that we were using for this. So we did some histology on the time course because the point here was to try to sort of nail down that elimination of the tumor correlated chronologically with presence of the virus. And so we can see here that in our vehicle treated, we really have no death within the tumor. When we add Zika by day 5, we're getting about 80% tumor necrosis, and by day 7, 95%. Whereas when we screen for NS1, so this is a specifically viral protein only present in permissive cells, by day 2 in the infected tissue, it is massively expressing. The virus has passed everywhere. And by day 5, even higher and retaining. So what we've seen in culture actually is something very similar. Typically speaking, we don't see cell death until about day 3, and most cultures are completely eliminated either by the end of day 3 or day 4. And so that tended to correlate here with the tumors as well, the time course. So by day 2, you have this massive infection within the tumor, but really the cell death only occurs following that point. Okay, so since the whole purpose of this is translational, as far as I'm concerned, everything else that we've shown, that's all good and fine from a basic perspective to give us a background, but the bottom line is survival advantage. So survival advantage is answering the question of, does this therapy actually help the host? Because if it doesn't, then this is all sort of academic, right? And so a survival advantage, these are Kaplan-Meier's, and basically what we're doing is introducing the tumors, then introducing the virus, and seeing, is there a benefit? And so these are IMR32s, this pretreatment MICAN amplified. This is all SMS CAN, also a pretreatment MICAN amplified. SMS CAN, that post-treatment metastatic, and then the SMS CAN-R, which is the matching patient to the SMS CAN, post-treatment MICAN amplified. And what we're seeing is consistently the same thing. So mice that are opted out are opted out at a tumor size of about 2,000 millimeters cubed. So that's the FDA standard for elimination of a preclinical mouse. That size is about the size of the mouse's head. So it's generally accepted that if the tumor gets to this size, there's no way that the mouse's immune system is going to self-eliminate. So once it gets to that size, the mouse is no longer considered bright alert, responsive, healthy. And so by IACUC standards, we're required to remove them from the study. And so what we see is that consistently, when we introduce Zika, we can actually eliminate the tumor, it doesn't grow, and the mice survive. So some of these studies, again, with like the SMS can, they go out as far as 100 days. That's a slower growing tumor as a pre-treatment. But all of them actually we continue with the Zika treated for another 4 weeks following, and again we generally don't get recurrence. We did lose one mouse in the SMS can, and again, because this is a direct inject and we're only doing a single dose, if we miss the target, then the virus doesn't go where it needs to. What we've discovered is actually within the mouse, since the mouse's interferon response is so effective, if you miss the target, the interferon response will turn on, the mouse will self-eliminate the virus, and it will never get to the tumor. So as a treatment approach, it really, you do need to be reasonably close to the target within the bed of a resected tumor, that sort of thing. Okay, so we wanted to also know, we've done cell lines, that's all good and fine, but we really need to know within patient samples, is CD24 really as overexpressed as we believe within the cells? And what we did was do a tissue array, 64 different neuroblastomas. So these include retroperitoneal, mediastinum, pelvic cavity, and adrenal gland. So that accounts for about 92 to 94% of all neuroblastomas are in one of these four places. And then we screened CD24 expression on them and found that on average, CD24 was about 450% higher expressed in neuroblastomas than they were in peripheral nerve. So generally speaking, barring pelvic cavity, which was not too well expressed, all of the others are significantly higher. Mind you, pelvic cavity only accounts for really about 2 to 4% specifically of those neuroblastomas, so it's not a huge percent. So well over 85% of the neuroblastomas are CD24 expressors and significant, and so that prognostically would be beneficial for this therapy. Okay, so since our interest here really is preclinical and translational, we really have to look at how clinically this therapy would fit into the model of treatment. And so, as far as neuroblastoma is concerned, induction therapy, which is the first step, really is I mean, again, this is basic chemotherapies and potentially can include surgery, but that really only accounts for 15% of success. In other words, 85% of patients are not responsive to induction therapy. So then it follows up with consolidation therapy. This includes radiation and much more aggressive chemotherapy. Cisplatin, by the way, which can cause deafness. It actually severely can cripple muscular growth. It can cause kidney and liver failure. So these are the kinds of chemotherapies that get used. They're pretty severe. But unfortunately, consolidation therapy only accounts for a total, mind you, not an additional, of about 40% that respond. So again, at this point, 60% of patients haven't been responsive to either. Then they go to post-consolidation, and to be honest, that's not a lot more effective because overall survival is only 40 to 50%. So post-consolidation therapy accounts for 0 to 10% improved survival rate. That's it. Okay, so the vernacular now is including a term called bridge therapy. And the argument is that the real window of opportunity is somewhere between induction and consolidation therapy. And so if the induction therapy doesn't work, which is by comparison the least abrasive approaches, that's really where you want to step in with new therapies that could be have a strong impact early on. Because once consolidation therapy is done, odds are good nothing you do after that's going to make much difference. And so we see that that's where Zika virus would fit in, following surgery, direct injection into the bed or into smaller tumor masses that surround that are too inconvenient for surgery. And since a lot of this tracks to the spinal column, there are a lot of places in the spinal column that surgery is not viable. Okay, so one example of that we've done here at Nemours was our bedside to bed example. So this was a patient actually at Nemours who had late stage 4 neuroblastoma, high risk. She was treated significantly. Unfortunately, she did not survive. But in the initial therapy, the induction, they did do surgery, remove the section. We had IRB approval. We acquired a sample. Within 2 hours of resection, I was there with pathology. They removed the sample. I raced to the lab, put it in dishes, and added virus. And by the end of the week, so we did a 5-day, what we found, as you can see here, so the bright field, and then that's a cell tox assay, so quantitative. Within 3 days at medium to high dose, we could completely eliminate the tumor. Within 5 days, we could eliminate even at low dose. Compared to background. So you can see in the bright field, the dark spots basically are indicating necrosed tissue, which is sticking to it themselves, glumming up in the middle, whereas the uninfected, it's largely obviously still alive. And so what we found was that on this patient sample, Zika was able to eliminate the tumor mass within 3 days. So that's a ex vivo example of bench to bedside to bench. Okay, so another cancer I want to talk about that we've also been working on is DIPG, so diffuse intrinsic pontine glioma. So this occurs in the pons, which is the base of the brainstem. There's only about 300 to 400 cases a year. It's all pediatric only. This is a developmental tumor, so typically by the age of 6, if you haven't had it, you're not going to. The problem with DIPG is it's completely untreatable, 100% mortality rate. There is no therapy for it. In fact, once it's diagnosed, typically speaking, children will be put on palliative, and 90% mortality in the first year, 99 within 2. So there is no therapy that exists. Sometimes they use radiation, but really no evidence that that's necessarily effective. So we've been working on DIPG with Zika for a little while. And what we found in cultures is that we could completely eliminate it within about 3 to 4 days, even under low dose. And so now what we're working on is an intracranial model. So there is no established model for DIPG. It's rare enough and doesn't have enough attention. We've done sub-Q models and they've been successful. So this is a collaboration with Sigrid Langhans in Delaware. And what they did is introduced our DIPG cells labeled with luciferase. And here you can see the spot in the brain of the mice, and they were asymptomatic effectively with the tumor anyway. So we're going to move on the next step to doing the virus injections and see if we can't, we can't eliminate the tumors within the cranium. So this is our team. Here we are doing one of our lab team lunches at Chili's at the end of the meal. So you can see we're all smiling because we're full and pretty satisfied. I'm a chips and salsa fan myself. These are some of our collaborating groups here, both in Orlando and elsewhere. And I'll take questions.